Welcome everybody to OA Week 2024. We're going to wait just a few more minutes to give people a chance to sign in. Thank you. Welcome to OA Week 2024. We're very excited to have you. And we're going to kick off OA Week with the South Asian Hub for Ocean Acidification. So just a few words about Ocean Acidification Week. First of all, I'm Katarina Schuh. I'm with the Intergovernmental Oceanographic Commission, and I'm a member of the GOA-ON Secretariat. You might have seen emails from me. And I want to just introduce you to OA Week. So OA Week is sponsored by 4 agencies. First of all is GOA-ON, obviously, the Global Ocean Acidification Observing Network. Second is NOAA, the United States National Oceanic and Atmospheric Administration. Third is the IAEA-OAICC, which stands for the International Atomic Energy Agency, Ocean Acidification International Coordination Center. And last but not least, the IOC of UNESCO, which is the Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific, and Cultural Organization. So GOA-ON. I'm sorry, I have a slight lag with my slides. Give me a second, please. Sorry. Sorry, slight technical issues on the first session of the day. Here we go. Excellent. So back to the GOA-ON, which was funded in 2012. And I just had a handful of members. I think 30 members or something were on the list for the first meeting. Since then, GOA-ON has grown tremendously. We now have over 1,200 members from 114 countries. We also have 10 regional hubs as of a week ago, which span across continents and oceanographic regions. And we'll be hearing from most of them this week, including the one we'll be hearing about just now, the South Asian Hub. So if you're not a member yet, we invite you to join GOA-ON by visiting goaon.org. OA Week started in 2020 and returned in 2021 when all events and conferences were postponed due to the COVID-19 pandemic. Following the successful in-person meeting at the Ocean and High CO2 World in 2022, we had another successful OA Week 2023, and apparently the community liked it so much that we're bringing it back this year again to maintain the momentum around ocean acidification research and to provide a virtual platform for the ocean acidification community to exchange their latest findings. And we're really thrilled to yet again have a lineup full of exciting ocean acidification topics and speakers from all around the world. A little bit of housekeeping. During the presentation, all participants are in listen-only mode, and you're welcome to type any questions into the question box at the bottom of your screen. We'll be monitoring all of the questions, and we have the chance to ask them after the 3 presentations we'll have today. You can also, if you like, raise your hand, use the raise hand button, and we will call on you directly. And with that, I would like to introduce Ms. Loke Baduri from the SAROA Hub. who will give you a little bit of an introduction into what the hub actually is about. So please take it away, Phoebe.
Thank you so much, Katharina. As Katharina has been pointing out that GOA-ON is spread, across all the continents that you can think of, and more than 114 countries, and the youngest one being in the Southern Ocean hub, which has just come up. So it tells us how important it is to understand our global ocean from the viewpoint of ocean acidification. South Asia Regional Hub on Ocean Acidification, SAROA, it came into existence way back at the end of 2021. It's one of the youngest hubs, but it's a growing hub. It has got a huge geographical expanse that spreads from Iran at one end and up to South Korea. There are more than 120 plus members. The SAROA Hub, has regular webinars. It also releases newsletter through which ideas and exchanges are being shared. It also involves the hub also participates in many international meetings related to ocean acidification, carbon cycling, and beyond. And last but not the least, as Katharina was pointing out, that OA Week is one of the most important events where South Asia Regional Hub members come together and highlight the activities on ocean acidification and implications for policy, coastal communities, and beyond. So today we have some exciting line of speakers. So I'll not take the spotlight and rather pass it to our speakers. We're going to listen from them. So over to you, Katharina. But before that, you're most welcome to join. You'll find it in the link for joining South Asia Regional Hub on Ocean Acidification at the GOA-ON website under the hub link. And if there's any issue, you can always write to us or write to the GOA-ON Secretariat. Over to you.
Thank you very much, Phoebe, for this excellent introduction, starting out strong. And with that, I would like to introduce your first speaker of the day. We have Amit Kumar from the KumarSathyabama Institute of Science and Technology in India, who will talk about marine macrophytes in the future acidified water. Amit, the floor is yours.
Yeah. Thanks, Catherine, for organizing, being a part of the organizing team for the OA Week. And thank you, Dr. Puneshlok Patri, for including SAROA in the OA Week. And it is really a happy thing that we are, starting the OA Week, and we are the first one to, initiate the talks. Before going further detail, let me share the screen. Just a moment. Okay, I hope my screen is visible. Can anyone confirm?
Looks good, thank you.
Yeah. So let us look what we've been doing in the field of ocean acidification, especially with the seaweeds. I am Dr. Amit Kumar, presently working as associate professor in Sathyabama Institute of Science and Technology, Chennai, and I am leading a research lab, that is marine biology lab, in the Center for Climate Change Studies. Before going into the topic, let me just give a quick introduction. I am based in Chennai, which is in Tamil Nadu, South India. And most of the studies, what we are doing is in the Kovalam beach in Chennai, as well as in Gulf of Mannar and Park Bay area in South Tamil Nadu. We've been looking into the marine biodiversity assessment in these two locations, and we are also involved in understanding how the species which are inhabiting in these two locations are responding to predicted climate change condition, in particular to ocean acidification. We are also involved in a number of citizen science initiatives in order to spread the awareness about the marine biodiversity and conservation through various different events. Now, coming to the topic, we know that atmospheric carbon dioxide is increasing at an unprecedented rate, at least in the last few decades or century, and that is leading to the increase in the global temperature. Now, that causes global warming. and also ocean warming. Another important aspect of the increased anthropogenic carbon dioxide in the atmosphere is that 1/3 to 1/4 of these carbon dioxide is being taken up by the by ocean, and that is causing changes in the or affecting their carbonate chemistry. Now that leads to ocean acidification, that is decreasing of the pH in the marine water. When I started working on the ocean acidification almost a decade back, there was an excellent meta-analysis paper which was published by Christy Croker in 2013, and thereby in that she showed that most of the calcifying marine organisms are severely or negatively impacted because of the ocean acidification. That was very much valid, because we know that in the acidified condition the cells decalcifies, and also at the same time the lesser concentration of the carbonate ion may be a limiting factor for their cell formation. If we look again into the same meta-analysis paper, we can see that especially in the non-calcifying algae or the seagrasses, that is the non-calcifiers, most of the studies till that time were very much limited, and that also those studies were limited to photosynthetic performance and the growth, and that was also understandable because carbon dioxide is a substrate for photosynthesis, so most of the studies were limited to that particular aspect till then. However, in the last decade, we have seen that a lot of studies have came and examined not only the photosynthesis and the growth of these fleshy algae or the seagrasses, but also looked into the physiological, biochemical, and the molecular processes as well. In the first 5 to 6 years during my PhD at Stazione Zoologica in Tordon, Naples, I have also studied Sargassum vulgare adaptation in the natural carbon dioxide vent, and we studied a number of physiological, biochemical, and the molecular processes which help the Sargassum vulgare to adapt in those high carbon dioxide, like atmosphere or the lower pH condition in the natural vents. However, upon coming back in 2018, we realized, or we saw, that not much of the studies have been happening, especially to understand that how the tropical seaweeds growing along the Indian coastline are doing under the acidified condition. So we started working on that. But before that, let me just give you a brief overview of the macrophyte, which is an umbrella term and which consists of seaweed, seagrass, and the mangroves. And we know that the macrophytes are very, very essential, not only from the ecological, but also from the economical point of view, right? So they play a very important role in the coastal environment. They contribute toward the primary production. They are very much important for the carbon sequestration. They form the base of the coastal food chain and the food web, supports numerous marine organisms, fishes, invertebrates, as a feeding, breeding, and nursery grounds for those associated faunas. They are also involved in the coastal protection and the nutrient removal. So any impact, if there is going to happen because of the ocean acidification on the macrophytes, it is not only going to be, like an organismal response that is going to be amplified to the coastal food chain or the food web, because as I said, that numerous marine organisms are associated with the macrophytes. In the last few years, we have also seen that a number of studies have came up which showed that the accumulation of the metabolites or nutrients has led to bioaccumulation and the biomagnification in the next trophic level in the coastal food chain and the food web. Apart from that, there are also studies which suggest, or which has shown, that there is a impact of ocean acidification on the consumer-prey relationship, or we can say like this algal herbivory or macrophyte herbivory relationship. So all those things may impact very severely the stability of the coastal food chain and the food web. So keeping that in the mind, we have started to study the impact of the ocean acidification on the tropical macrophytes. Some selected seaweeds and the seagrass, which are listed on the slide. we have brought them to the laboratory condition and then exposed them to the lower pH condition. And we started understanding that how the general physiological as well as the metabolic profiles are changing under the acidified condition. Going back again, that I will tell you the story of two of the seaweeds which we have studied in the last few years, and those were collected from Kovalam and Parkway in Rameswaram in South India. So when we started to study the seaweeds, we first looked into that what are the common different seaweeds along the Tamil Nadu coastline are available. Of course, there are more than 200 species, but there were selected 20 to 30 species which were very often finding it, and then decided that okay, we will start working on these particular seaweed. So first step what we did is that we started to think that whether these seaweed do have the carbon concentrating mechanism. Let me give you a moment of understanding that what this carbon concentrating mechanism is. So unlike in the air, in the sea or in the water, the carbon dioxide diffusibility is limited, and thereby causing the limitation for the aquatic macrophytes to perform the photosynthesis. So as a result, they have, or aquatic macrophytes have adapted or have evolved a mechanism of that is the uptake of the bicarbonate ion. That is the carbon concentrating mechanism, right? So a review which was published by Margaret Koch in 2013 showed that more than 95% of the aquatic plants do have the carbon concentrating mechanism. So we started from that point, and then based on the pH drift experiment, we evaluated that most of the seaweeds which we have collected from the tropical waters along the Indian coastline do have the carbon concentrating mechanism. Not only that, we took some of the selected seaweeds and also performed ion transporter inhibitor experiments in order to see that whether the algae which were already performing the like passed through the pH drift experiment does show any changes in the photosynthetic rate or not. So we indeed found, as you can see on the screen, that two species of the Gracilaria, that is Gracilaria debilis and Gracilaria folifera, in presence of the inhibitors have reduced the RETR max through the PAM when we analyzed, suggesting that these species do have the carbon concentrating mechanism. Now, of course, I cannot be able to speak for all the species in today's talk, so I'll be limiting my presentation today only for the Gracilaria. Now, Gracilaria, we know that it's a very widespread genus. There are more than 100 species worldwide available, and many of these species have the economic importance, such as the agar production. They are involved. They are even being consumed directly by the humans in the poultry feed. And many of these species do have the bioactive potential, which has been shown in the literature. So as in worldwide Gracilaria is being cultivated, there are many species even cultivated along the Tamil Nadu coastline. So two of the species which we have taken, that is a Gracilaria debilis, collected from subtidal waters of Rameswaram, and Gracilaria folifera in the intertidal region from the Kovalam. And we brought to the lab, we acclimatized it for almost 1 week. Post that, we exposed it to the lower pH condition through the carbon dioxide pumping. And this is the model of the experimental setup which we did. And after 2 weeks of the incubation, we measured the response variables. First is that how the growth, primary productivity, and the photosynthetic pigments are impacted. Second is that how the algae, general stress response of the algae is by looking into the oxidative status. And in that, we have measured total antioxidant capacity, antioxidant enzyme, and non-enzymatic antioxidants. And in the third, that we started looking at how the primary metabolites, in particular to amino acid and the fatty acid, is being influenced under the ocean acidification. And last, we checked the bioactivity, crude extract bioactivity against few of the pathogens. So as I said previously, that these species do have the carbon concentrating mechanism. But even in the literature, we have seen that the algae or the macrophytes, even if they have the carbon concentrating mechanism, they increase the growth rate, productivity, maybe because the carbon concentrating mechanism is an energy intensive mechanism, and as a result of which the algae may have, if they are exposed to the high diffusibility of the carbon dioxide, they may increase the growth, or they may start utilizing the dissolved carbon dioxide than the bicarbonate ion. That may be one of the reasons that we observed high growth rate under the acidified condition in both the species, and that was even supported by the enhanced primary productivity and the photosynthetic pigment. Now we know that if we look just at the photosynthesis and the growth, it is positively it's a positive response. But we also wanted to see if the algae is undergoing any sort of oxidative stress. So for that, we can see that the total antioxidant capacity was not changed under the acidified condition. Lipid peroxidation also did not elevate under the lower pH condition, and the battery of the non-enzymatic and the enzymatic antioxidant which we measured in both the species in the control and the acidified conditions showed comparable results, suggesting that the algae may not be undergoing the oxidative stress under the lower pH condition. Now, after the growth and the physiological response, we looked at how the metabolites are changing. So since the primary productivity was increased, we were expecting that the sugar also will increase, and indeed it was. We found that increase in the soluble and the insoluble sugar in both the species under the acidified condition. If we look into the amino acid, in general, there were more than 30% increase in both the species. Particularly, it was more important that the essential amino acid was enhanced in Gracilia dieblis, suggesting that the nutritional quality, at least in terms of the essential amino acid, is not going to be affected under the acidified condition for this particular seaweed. However, if you look into the fatty acid profiling, overall it did not show much of the change. But if you closely observe, the monounsaturated and the polyunsaturated fatty acid showed a decline of almost 10% in both the species. And now that's a reason to consider that if such thing happens in future, and if the algae is growing in the acidified condition, maybe the associated community who are dependent on these algae for the consumption or for the fatty acids for their reproductive and for the physiological processes will be negatively impacted. Since the sugar as well as the primary and primary metabolites showed increase, we haven't analyzed the secondary metabolites in detail, but we assume that could be the reason for increase in the bioactivity for most of the against most of the bacteria and the fungal pathogen which we have tested using the crude extract of the algae grown in the control and the acidified condition. So in a nutshell, if we see that after the 2 weeks of the exposure for at least for these two species, we do not see any negative response. Rather, we can say that it's a positive response under the acidified condition. And the result of this particular work, which I am presenting today, has been already published last year in Marine Pollution Bulletin. We understand that there are few limitations. For example, the study was conducted for 2 weeks. It is a single driver experiment, and we know that most of the time it is not that only one stress will happen in the nature. So over the year, we have also now a very well established facility, recirculation system facility, to conduct the multifactorial experiment. So maybe the experiment is ongoing with different species in the lab, and hopefully in the next OM meet, I will be able to give more detail on those experiments. But before to end the talk, I will just give you some of the ongoing experiment in the laboratory. We started seeing that how the acidification and the warming is affecting the algal palatability. So again, we went ahead with the Gracilaria folifera and then the Litorina species gastropod, which was associated with the Gracilaria. And we grow the Gracilaria under the different condition. As you can see that in the controlled condition, high temperature of 32 degrees Celsius and the pH 8.1, low pH and high temperature, at low pH and normal temperature under the different conditions. And we after a certain duration, we exposed, or we took the algae, we divided in two parts, one incubated with gastropod and one without gastropod. And as you can see, the preliminary result suggests that the algal palatability, or you can say that the feeding rate of the gastropod to this particular gassilaria is being impacted because of the different conditions which we give. So again, it's a very preliminary result. So that's why I'm just giving you some of the outcome, and more I'll be able to share in the future. The second experiment which is ongoing is on to look that if the seaweed can provide refugia to the associated or the nearby organism. For that, we have studied with the intertidal green seaweed, that is a Chaetomorpha antenina, and the associated fauna. So before to choose that with which species we will be working in this particular experiment, we did the temporal variation in the associated community for a year in this particular seaweed in the in Covalum rocks. And we found that, as you can see, that some of the common associated faunal communities and how it is being changed across the year, all those results. I'm not going in detail with that, but one particular highlight is that we found a novel species of the amphipod, that is a Protohylae covilongensis, which we named after the region from which we have identified, that is Covalum beach. So we found that this particular Protohylae covilongensis is abundantly present on the Chaetomorpha antenina. So we performed various basic experiments. As you can see that the outline I have shown you in the slide. We first took only the seaweed in an open container, in a closed container, seaweeds with the amphipod, without the amphipod. And also the second experiment, that we took the seaweed in one container, amphipod in the other container, we connected it or bridged it with a tube, and then as a control we connected it to the tube, but we disconnected the tube in order to prevent the amphipod movement to the seaweed. And as a result, what we have found, that the seaweed container, where the average pH, and the experiment was done with the normal pH of 8.1 and the lowered pH of 7.7. And you can see that even after 5 to 6 hours, we can see that the average pH of 7.5 rose to around 8.5 in the seaweed container. 100% of the survival rate was found in the amphipod with the seaweed containers, whereas 40 to 50% mortality were found in the amphipods if we are not keeping it with the seaweeds, right? And at the same time, in the connected container experiment, we found that 40 to 50% of the amphipod migrated to the seaweed container through the connected pipe. And in the connected container, the seaweed container pH raised to 8.04 after 5 hours, whereas the amphipod container remained at 7.5 plus minus 0.1 pH. So this further indicates that seaweed do have the potential to provide the refugia for the associated community. Again, these are very, very preliminary experiments, and the experiments are ongoing. So maybe in the next year in the OM meet, I'll be able to present more results related to this. So with that, I would like to thank the organizers for inviting me to speak on the OA week. And most of the experiment was conducted as a part of the PhD thesis of my PhD student, Dr. Vinu Ganesh. So I'd like to thank him and all the other lab members who were involved in the experiments. And the research was funded by SCRB and Satyabama University. So I'm open for discussion if you have any questions. Thank you.
Thank you very much for this excellent presentation. We will have time at the end of our 3 sessions to have questions. But however, if you have questions, please feel free to use the Q&A box to type in your questions, so that the presenters can see them and can already think about them potentially. And with that, I would like to introduce our next speaker, who is, give me 1 second here, already waiting to hit the screen. We have, excuse me, I'm really having some technical issues this morning. We have Dr. Kunal Chakraborty, and He's with the Indian National Center for Ocean Information Services, who will be talking about understanding the present status of the Indian ocean acidification and its driving mechanisms. And with that, the floor is yours.
Yeah. Thank you, Catherine. I hope my skin is visible to all of you.
Looks great, thank you.
Thank you. Okay. So, good morning, good afternoon, good evening to the participants. As already introduced, my name is Kunal Chakravarti, and I'm going to talk about our present understanding or the present status about the Indian Ocean acidification and specifically the mechanisms which is actually driving this acidification. And I'm working as a scientist at INCOIS, which is under the administrative control of Ministry of Earth Sciences. And thank you, the organizers, for providing me an opportunity to showcase our recent findings in this meeting. Thank you. Okay, so a brief outline of this talk is something like this. I'm going to take you first about the known knowledge, or rather the present status of Indian Ocean acidification. Then I will take you to the recap 2, which was concluded in 2023. It's kind of a assessment, regional carbon cycle assessment and processes that is conducted by a global community. And then I will take you to the modeling system that is developed at INCOIS. And based on the outputs of these specific models, I'm going to show you the acidification status in the different subregions, specifically the trends and the driving mechanisms. And then I will try to link the acidification with the climatic modes like El Nino and the positive Indian Ocean dipole and all. And then finally, the summary of this specific talk. Okay, so start with, it is needless to say that ocean plays a very important role in mitigating this global climate. And specifically, if we talk using numbers, the global oceans is approximately sequester 30% of anthropogenically emitted carbon per year. That is the estimation that was provided by Lecure in 2018. And the most important fact of this is, although tropical Indian Ocean is very smaller compared to the other basins like Pacific Ocean or the Atlantic Ocean, as per the available estimates, almost 16% of the total ocean carbon sink is actually taking place from the Indian Ocean. And as a result of which, specifically the Indian Ocean, which is become or rather the surface waters, pH is basically decreasing, and Indian Ocean is become relatively acidic. Obviously, there are specific subregions or the pockets which is happening in a more faster manner, whereas there are few places where it is not as fast as faster than the compared regions. However, if we talk about the CMIP-6 models, as per the estimates available from the CMIP-6 models or the global climate models, there are estimations which talks that the Indian Ocean will be acidified, or the pH will be reduced by almost 0.3 to 0.4 unit by the end of 21st century. Now, if we specifically talk about the Indian Ocean or the estimates, those are actually available from global community, then we can see the Western Indian Ocean, which is actually acidifying in more faster manner. And according to Seus et al., during 1960 to 2010, almost 0.07 pH units got reduced in the Western Indian Ocean, rather in the western part of the Arabian Sea. Now, Based on the available observations, there were several studies that were actually estimated the acidification for the last 3-4 decades. One of such study is the Loveset 2015 that came out with an estimates of like 0.027 per decade. There's an decrease in the pH, Indian Ocean pH. Whereas there are two recent studies. One is Chow et al., another is Ma et al. According to those studies, the decreasing rate is not as much as 0.027. However, the rate is, according to their studies, is like 0.015 to 0.017 per decade. But here we need to remember that the number of available observations, specifically in the Indian Oceans, are very limited, and that too in the spatial scale as well as in the temporal scale. So the ML, machine learning based methods or the algorithm, those are applied to develop this kind of observation based reconstructed data product that comes with an large uncertainty, and that is actually visible specifically in the Indian Ocean. Then if we talk about the ocean general circulation models, in the 2023, there was an assessment that was completed by the global community under RECAP 2. And under that specific process, it was shown that the regional models are actually doing good compared to the global models in capturing the spatiotemporal dynamics of PCO2 and the subsequent change in the ocean pH. So if I briefly tell about the recap assessment, it is actually meant for entire global carbon budget. However, if we talk about the ocean Pacific part of the recap 2, a global consortium of partners, they have actually come up with their model simulations, observation-based data products, and all possible observations that are used to come up with a best estimate of ARC CO2 fluxes and the associated variables which is regulating these ocean carbon cycles, specifically how good they are capable of simulating these specific variables in the different parts of the global oceans. So one such consortium partner team of partners consists of for the Indian Ocean. And under that specific on this specific chapter, they came up with the first assessment during 2013. And during that time, it was concluded that none of the global models were capable of simulating the Indian Ocean carbon cycle. However, in the recent assessment, which is after almost after a decade, which concluded in 2023, it was demonstrated that the spatiotemporal variability of CO2 fluxes is better captured by the high-resolution regional models that are specifically meant for the regional oceans than the general global models. So based on that, what we did actually in 2023, in the recap assessment, inquires participated by developing a regional ocean model, which are capable of simulating these surface ocean PCO2 and pH. And you can see in this slide, the domain which is highlighted using this blue color box, which actually encompassing the Indian Ocean that is provided by the recap to community. And the different colors are actually provided for the different oceans. And for each specific basins, there are specific chapters. They actually came up with an assessment of the improvements that were carried out from 2013 to 2023 and what kind of drawbacks are there and how we can actually make improvements further based on this kind of an global assessment process. So typically this slide is actually providing you the model configuration details. I'm not getting into this more. However, let me just tell you here, we have actually used for the assessment the model simulated outputs from 1980 to 2019, that is last 40 years. And it's a coupled ocean ecosystem model. So it's actually simulates ocean physics as well as the ocean biogeochemistry at the same time. And in order to validate how good it is doing, we have actually used this Bay of Bengal ocean acidification based mooring that is located at 15 degree north, 19 degree east. And then SOCAT, there is a SOCAT database which is available. And it's actually mostly consists of SIP-based observations of PCO2 and apart other regulatory variables. Those are used, and we use two observation-based data products. One is ocean soda ETIG, and then CMIMS data product. So these are the specific data products are used to come up with a comprehensive assessment to understand the changes in the Indian Ocean surface pH. So if you can see here, specifically, this particular slide is showing the model, how good it is able to capture the SST and SSS variability in the Indian Ocean. And you can see these are the data sets, these are available along the cruise track. So all last 40 years, sea-based observations were collected, and then we have made this assessment. And you can see the model is actually capable of simulating these SSS and SST very well, and the correlation and specific RMSE is actually available in the table itself. Now, if we talk about how good it is doing with respect to SOCAT-based observations, so the first panel, it is actually showing how good ROMS, that is the model being used for simulating this surface PCO2. And below there is another model output we have used that is developed at IITM Pune. And that model is an offline model, which is not simultaneously simulating the ocean biogeochemistry, but it's an offline model which is capable of capturing the ocean biogeochemistry, and it's a global model. So at the same time, we made use of a global model to see how good it is actually doing. So based on the statistics, you can see here as well, the regional model or the high resolution regional model is actually doing better job compared to the offline global biogeochemical model. Then we wanted to see, based on the Bay of Bengal ocean acidification mooring, how good it is actually capturing the seasonal cycle and the interannual variability. So from this specific slide, you can see the ROMS that is showing using the red color. In case of PCO2 and pH both, the seasonal cycle is well captured as seen from the Bay of Bengal ocean acidification mooring data. Whereas OTTM, although it is captured the seasonal cycle well, but the magnitude as well as there are phases where it is actually not coming closer to the mooring-based observations. So after this, then we wanted to see, based on the model outputs, what are the driving mechanisms that controls the changes in the surface ocean pH. So that's a very commonly used equation available in the literature, which actually describe that the changes in the pH is nothing but the changes as a function of the changes of DIC, alkalinity, SST, and SSS. So what we did is we have used the model simulated DIC, alkalinity, SST, and SSS, and then we came up with a construction of pH using this equation that we called the control pH. After that, what we did, let's say I want to do some sort of sensitivity experiments. So in order to do that, what we did first, we have removed the trends from SST. So we basically detrended the SST. However, all other variables like DIC, alkalinity, SSS remains same. So using the detrended SST and other variables, again, we reconstructed the pH. That we call as sensitivity with respect to SST. Now, if I subtract the control pH with the pH which I am getting through sensitive SST, it's going to give me the contribution of temperature in the changes of pH trend. So iteratively, this process were continued with respect to all other variables, so that we can come up with a clear estimation how much those specific variables are contributing in the changes of or changes in the trend of pH. So the next table here you can see categorically, although we have done it for several other subregions of the Indian Oceans, but Bay of Bengal and Arabian Sea, these are the two most important ecosystems in the Indian Oceans. So what we did, we categorically checked how the changes are taken place in the Arabian Sea and Bay of Bengal. So from this specific slide, you can see in the Arabian Sea, the changes that is being taken place by temperature is like almost 15%, that is 14.79%, whereas alkalinity changes is 5%. And DIC is actually dominating the changes in ocean pH trend, that is almost 80%. and salinity, it is not having very it is not significantly contributing to the changes in pH trend. It is almost 0.28%. So if we just add all the things, so the trend that is not explained is 0.52%. So from that, we can conclude that specifically the decomposition that I described in the previous slide, it is actually doing well. Almost 99.5% of the variance that is being explained based on these specific contributions. So similarly, you can see the same numbers from the Bay of Bengal here. So if we compare both the tables, the most interesting fact is that in the Bay of Bengal, alkalinity has a buffering impact. So it is basically reducing the decreasing pH in the Bay of Bengal, whereas in the Arabian Sea, it is actually enhancing the pH trend. So that's the most contrasting behavior that you can observe from these specific tables. Apart from that, obviously in the Bay of Bengal, the DIC is dominating to the fullest extent, whereas the DIC and temperature both are having significant control in the Arabian Sea trends. Now, why it is happening, the contrasting case we are getting from the alkalinity. The most important, or the what we hypothesize, is something like the Bay of Bengal. It is lot of discharge, these glacier waters and perennial rivers. These are discharging in the Bay of Bengal, like Ganga, Brahmaputra, Yamuna, and all. So these rivers, these are coming with very high alkaline waters into the Bay of Bengal. And that particular water is actually coming as a significant amount in the Bay of Bengal. So that's specifically one reason which is actually in basically buffering the pH trend in the Bay of Bengal. Now, to proceed further, if we see the slide, how these specific changes are linked with the climatic modes. What we did is something like we did EOF analysis. It stands for empirical orthogonal function analysis. So we have 40 years of data. We just we have calculated the anomaly, pH anomaly first, and then we did the EOF analysis. And the first panel is showing the EOF1 and the second panel is showing EOF2, whereas the time series is showing the correlation and the correlation with the first principal component with dipole modes index and NINO 3.4 index. And the second mode, the second panel is showing the principal component 2, the correlation of principal component 2 with DMI and NINO 3.4. Now, if we try to explain the figures or the specific trends that is being observed here, you can easily see that the EOF2, it is a typical pattern of positive IOD. And if you see the EOF1, here you can see from the correlation as well as the color variation that is taking place in the EOF1, that ENSO and the IOD both are actually contributing to the changes in the EOF1. If you see the correlation, it is clearly visible. The DMI is actually correlating 0.37%, and NINO, that is basically representing the ENSO, which is dominating, that is 0.52. Whereas in case of PC2, DMI is actually 0.30, whereas NINO is not having any impact. That is, ENSO is not having any impact in the PC2. So from that conclusion, what you can see, the ENSO and the positive IOD, it is actually enhancing the Indian Ocean acidification. Now, to proceed further, if you see this slide, where what we have shown here is how last 4 decades the changes are taken place in the Indian Ocean. And for this, we have used these observation-based data products as well. So ROMS, it is represented using the red color. CMIMS, it is using the black color. Soda, that is ocean soda, it is using the blue color. And the green color is showing the OTTM. So for the Arabian Sea, you can see how the per decade change has been taken place from Arabian Sea, and then followed by Bay of Bengal, then equatorial Indian Ocean, and then the overall entire Indian Ocean. So from this left-hand side, you can all see that OTTM is actually deviating a lot compared to the ROMS-based simulation and other observational-based data products. Whereas the right side, if you can see, the contribution of all these drivers in regulating the pH trends are shown. So from 1980 to 1989, that is the first decade, then followed by other 3 decades are shown. So most important fact here is that in the 2000 to 2009, that is the specific decade where in the EIO region, if you can see, the alkalinity has a very severe impact compared to all other decades. However, if we see the changes throughout the Indian Ocean and in terms of drivers, mostly the temperature and DIC, these are the prominating driver, and they are counteracting their impact in the pH trend. Whereas the contribution of temperature in the pH trend has been changing or has been increasing for the last 4 decades. Now, in summary, what we have seen from that analysis is something like the rate of change of the decline pH in the Arabian Sea and the Bay of Bengal is like 0.014 plus minus 0.002 unit per decade. And that's kind of an estimate which is also showing by the observational based data product that we have used in this specific analysis, that is CMIMS and SODA. And the trend of dissolved inorganic carbon primarily drives the Indian Ocean acidification trend. The increasing anthropogenic CO2 uptake by the ocean dominantly control the pH trend in the ASBOB, whereas ocean warming also controls mostly in the AS, however, than in the Bay of Bengal. And the most contrasting fact is that the changes in TA contributing to the enhancing pH trend in the Arabian Sea, whereas it has a buffering impact in the Bay of Bengal, which is essentially suppressing the pH trend. And then El Nino and the positive Indian Ocean dipole leads to an enhancement of the Indian Ocean acidification. So it's our, this study is recently got published in Global Biogeochemical Cycles. So for more details and the analysis, probably if someone is interested, they can refer to this article. And to acknowledge, apart from my Indian colleagues, like my colleagues from INCOAS as well as NIO, there are colleagues, French colleagues who were involved in this study, like Nicolas, Marion, and Frédéric and Claire. They are part of this specific study. And in terms of funding agency, Ministry of Earth Sciences, Government of India, they are the major funding agency for INCOAS. And apart from that, the study is funded by CMIMS to Marion is funded by CMIMS, and SOCAT data being used, which is publicly available. So all data are actually publicly available, and these are also listed even here as well as in the paper. If you can, like you'd like to make use of that data, these data can also be used from that. Even our high resolution ROM simulated data that are also archived in Genodo, and if you can, if you want to use that data, that can also be downloaded and can be used. Now, what would be the future course of action, or what are the future steps that we are actually pursuing right now is like that. This is the last slide where we have seen is like now, like in historical stage, or rather the past climate variability of ocean pH that we have already studied, and it is already established that there is a clear change in Indian Ocean pH. And based on CMIP6 outputs, it is also projected that there will be a decrease of 0.3 to 0.4 unit of pH in the Indian Ocean. So those CMIP6 projections are actually associated with lots of uncertainty. And what we are right now doing is we are developing high resolution regional future projections of Indian Ocean pH. So these CMIP6 models will be used, and then those models will be further downscaled and bias corrected. And then it will be taken as a high resolution regional model. And then probably we will come up with a couple model based projections. So this particular work we have been carrying out under Deep Ocean Mission. There is a project by Ministry of Earth Sciences where INCOAS is actually leading on the development of OCCS, that is Ocean Climate Change Advisory Services. So under OCCS, we have been developing these high resolution regional future projections. So hopefully by the end of 2025, we'll be able to have the regional future projections of ocean pH along with other BGC variables. Thank you. Thank you for your attention. That's all from my side.
Thank you very much for this very detailed presentation. I'm sure there's going to be a bunch of questions.
Welcome.
And with that, thank you so much. With that, I would like to introduce our next speaker to the webinar. We now have our colleague, Nabanita, who is from the Shajal University of Science and Technology, Bangladesh, and I apologize for my absolutely atrocious pronunciations. And Nabanita will be talking about the nano-DOP monitoring activities in Bangladesh. So please take it away, Nabanita.
Thank you, Catherine. Hello, everyone. Good afternoon from Bangladesh. Today, I'm going to talk about hello, everyone. Am I audible?
You look good, and we can hear you very well. Thank you.
Okay, thank you. So I'm master students from the Department of Oceanography, Shahjalat University of Science and Technology, and I'm also a nano research fellow of the NanoDope Global Project. And today I'm going to talk about one of the sampling station of NanoDope project that is in Bangladesh. So the title of my talk is NanoDope Bangladesh Environmental Monitoring in the Meghna River Estuary. So actually, what is NanoDope Global Project? So this is a global study of the coastal deoxygenation, ocean acidification, and productivity at selected sites. So we all know that the environmental conditions throughout the world ocean are changing in response to fossil fuel emissions and greenhouse warming in the atmosphere. And climate change and high carbon dioxide levels in the atmosphere are series of changes in the ocean conditions, including increased temperature, decreased oxygen level, and also acidification. So continuous monitoring at specific locations, specifically at the coast of developing countries, are very necessary for forecasting and for warning operations. So NanoDope Global Project is actually establishing a network of coastal monitoring stations. This is taking advantage of the nano members global distribution, because this project is run by the nano member. And this project was begun in April 2017 as the continuity of the previous regional observation project. And this project is funded by NFPO. So these are some of the sampling activities of this NanoDope Global project. And the general objective of this project is to support in situ measurements of selected essential oceanographic variables at participant study sites. Also to compile data submitted by the participants into nano global database of selected essential oceanographic variables, and support comparative analysis among study sites, focusing on the levels of the productivity, acidification, and also deoxygenation. And one of the objective is to provide capacity building opportunities to the project participants on marine data management, observations, and data analysis methods through webinars and also workshops. And last but not the least, to initiate and promote citizen science and outreach activities to raise awareness about the importance of the marine environment and the threats facing it. So this is the geographical distribution of the of this project, and at this moment we have 36 sampling sites in 18 countries of Africa, Latin America, and Asia. So today's talk is focused about the sampling sites, or nanodope sampling sites, Bangladesh. And you can see here, so this is the sampling station, Meghna River estuary, which is located in the central coastal zone of Bangladesh. And this sampling site was included in this project in December 2019. So currently, the environmental variables, what is continuously measured from the Meghna River estuary, includes temperature, salinity, pH, DO, chlorophyll, Midisecki depth, nutrients, specifically silicate, nitrate, phosphate. So we also have some mandatory variables. That means we have to measure at least temperature, salinity, pH, DO, chlorophyll, so that we call these mandatory variables. And from August last year, we also included Secchi depth as a mandatory variable, because that time we received Midisecki disk, which is a very portable device to measure Midisecki depth from the coastal area of the sampling stations of nanodope project. So this sampling, we actually do the samplings on a monthly basis, and the sampling depth depth varies from 0 to 8 meter depth. And from December 2019, we collected approximately 260 data points of the essential oceanographic variables from Meghna River estuary. So there are some of the time series data of the essential oceanographic variables. We can see here in these box plots. These are from December 2019 to September 2024. And we can see that the salinity range is quite low. That varies from 4 to 9 PSU. This is because Meghna River estuary actually receives, which is situated in the central coastal zone. So this receives massive freshwater input from GBM river system, that is Ganges Brahmaputra Meghna river system. So this dilutes the salinity in this region. And also during monsoon season, freshwater inflow increases due to heavy rainfall in the upstream catchment area, that also further lowering the salinity level. And we can also observe the gradually increasing trend in the DO measurements throughout a year, where we observed a higher oxygen level in the winter season, and a lower level in the summer season, which actually related to temperature and biological activity, and also organic matter input. So these were the scientific observations, and we also have some other activities. For example, the training program organizations for students and for early career professionals, so that they can learn nutrient analysis, plankton analysis, and other measurements of the oceanographic variables. We also do outreach activities. For example, we organized a workshop on plankton observation using Foldscope. This is a paper microscope, and this is very lightweight and efficient and low cost portable microscope. So we actually organized this workshop for high school students. During this program, we like students were introduced to these instruments, and also they learned how to use it to examine water sample collected from the coast. We also conduct citizen science activities. We try to monitor systems in a cost-effective way, and also we can make a wide coverage of data. So here we can see the user interface of the C4SAM mobile application. This mobile app was developed by the Bangladesh representative, and this app is called NanoDope C4SAM. So C4SAM actually stands for Citizen for Coastal Ecosystem Monitoring. Using this app, people can take photographs of resources, of any resources from any locations. If there is no internet connection, this isn't any issue, because this app automatically capture and store location and photographs, and whenever the mobile is connected with the internet connection, the data is transferred to our database. So in this way, we are actually trying to monitor plastic pollution and also marine resources in the coastal region. And this initiative is actually piloted in the Bangladesh coast, and now sampling sites from African countries are also using this app for their monitoring purpose. So at this moment, we are actually doing coastal ecosystem monitoring. We are doing capacity building activities, outreach activities, citizen science activities at the Meghna River estuary, and we are actually like continuing this monitoring activities. And our next step is to collaborate with another project of POGO, which is called SAGITA. This actually, like, this is a citizen science project, and under this project, a Russian representative developed a low cost temperature probe, and this can actually measure temperature profile very efficiently, effectively, from surface to bottom, and it can operate up to 200-meter depth. So this is how the sampling sites of Bangladesh is working, and they are actually contributing to this global research project. So this project, which is actually monitoring the coastal ecosystem across the developing countries in the Africa, in Asia, and Latin America. So that's all. Thank you very much.
Thank you so much, Navanita. That was a fascinating insight into some very interesting science. On that note, we also have a technology gaps and new science tools session happening later this week that you might be interested in personally. It's happening on Thursday, and you can find the link on that in the GOA-ON website. And with that, I would like to invite all of our presenters back. Please join me. We will now take questions from the audience. So audience, please don't be shy. You can either use the Q&A box to type in your question, or if you prefer to ask your question directly, please use the raise your hand button, and we will call on you to ask your question directly. Anybody want to go first? I know it's been quite the dense start. There's been a lot of information in these last 3 presentations. So maybe I will start with a question. I was wondering, because you've looked at, this is to Dr. Kumar, I'm sorry. You looked at these algae. Was there a reason you chose those particular organisms? And would you add other potential keystone species to your studies in the future?
Yes, so I presented 2 species today, but we have already studied 5 species of the seaweeds, and at least to the photophysiological response of 6 different seagrasses. But those results I haven't shown today. And moreover, with the seagrasses, we are also performing long-term experiments now with one of the other PhD student. So yeah, and the reason to choose those species was first was they were abundantly present in the study location. So sometimes it is easy to perform the experiment if the organism are there in the coast, easy accessibility. Second is that there were a lot of associated community with one of the Gracilaria species. So again, we wanted to have those species which are ecologically important as well. And as I mentioned that this Gracilaria is economically also very important species, and a lot of cultivation is happening across the world, including along the Tamil Nadu coastline. So considering all those, we chose those two Gracilaria species. But we have studied Ulva, we have studied Chaetomorpha, and few other species as well.
Thank you for that specification. I'm always very interested to see what organisms are chosen for what reason, because obviously there are reasons. However, sometimes not being from this region, the organisms obviously are not as familiar to me as they are to you. And so Nabanita, so we have heard about this being a whole network of different stations that work in this project. Is there an exchange that happens on a regular basis, as in do participants in this exchange on what worked, what didn't work, what their problems are, their challenges potentially, and how they've overcome them?
So, so in this project, there are 36 sampling stations from 18 countries. So, we are actually working quarterly. Like in every quarter, we deliver what we conducted, like what we have done during this 3 months period, like what we have done for the citizen-based activities or for scientific like measurements and capacity building activities, like all the things we conducted within that 3 months period. We like shared this with the coordinator and also to the nano fellow for conducting further analysis and for storing this data to that database.
Excellent. And just you mentioning database brings me to my next question for our third speaker. You have a very extensive collection of data there on ocean acidification from a region that is globally quite undersampled, and I would not be doing my job properly if I didn't ask, are you planning on submitting this data to the SDG 14.3.1 indicator? I'm asking because basically the IOC, the Intergovernmental Oceanographic Commission of UNESCO, is we're the custodian agency for this indicator, and we have just sent out again a letter to all member states and to our GOA-ON partners. And this is a region where the data is very sparse. So we do know there is studies being done, because we do know that you are all doing excellent work. But I was wondering if there are reasons the data hasn't made it into a global database yet.
So yeah, as this is from a global project, so Bangladesh is one of the sampling sites. So this data is included with that global project, and actually with the help of the NanoDope coordinator and also other members, we are actually planning to share this data, and also we are planning to available this freely available this data to people. So yeah, we are planning also to share this OA data to the SDG 14.3.0 portal.
That's excellent news. And the same question to Dr. Chakraborty, because you have more parameters than I have seen in a long time from your study area. So that's a very, very impressive data set. And I know that there are sometimes limitations into what you can share due to publications pending, but would you be interested in willing to share this to a global audience?
So basically in our study, we generated these model simulated outputs for 40 years that I will be happy to submit to the community. And in fact, it is available in Genodo. So anybody can go into that link and download that. As long as institute data are concerned, we mostly use this SOCAT database, which is freely available, and Bay of Bengal ocean acidification mooring based data that is also freely available. So model simulated data, if you ask me to upload somewhere, I will be happy to do that. That's not a problem. Anytime I can do that.
Brilliant. That's excellent news. We will be sharing a recording of this session. So if people haven't been able to catch the list of the resources that you had during your presentation, they will be able to go over this during the recording, which will be available on our YouTube channel, the GO-ON YouTube channel, very soon. And I am really wondering if there are any questions from the audience, please. Don't be shy. I'm just assuming you all did such a great job in presenting that everything is clear. And there are no more remaining questions. Professor Bajuri, do you have any questions you would like to ask your panelists?
I think you have already asked, but I have been bombarding, writing in the mailbox, chat box, that if there are ways to submit this data, because I think, yeah, we are seeing a lot of this new data coming out, probably 14.3.1 portal, if those gets uploaded, will be very, really nice for this data deficient region. I just have one quick question for Amit. Amit, are you what is the depth of these sites where you are looking?
So these are shallow, I mean, not very, hardly from 1 to 3 meters at the max.
Okay, thank you.
So these are shallow waters, subtidal system, basically. Yeah.
Thank you.
Excellent, very interesting. So you seem to have a shy audience. Maybe they're just warming up since Away Week only just started. But I would just like to thank you all very much for being part of Away Week, for kicking us off on a high note. This has been really excellent. And as I said, the recording is going to be available soon. And we would encourage you, if you're not yet a member, to join GOA-ON, participate, be a part of this community. And also, we are currently looking for new members to join the GO-ON Executive Council. So if that is something that you would like to do to shape ocean acidification and to shape the Global Ocean Acidification Observing Network, then please join us. And with that, I thank you all again. Excellent presentations. Thank you so much for teaching us more about the science in your region. Thank you very much. Goodbye.
A pleasure. Thank
Thank
you.
you, Kathy.
Thank you.
Thank you.
Thank you, Katharina. Thank you, panelists. Thank you. Bye.
Thanks.